Hello, thank you for coming to see my poster. Today I'll be presenting about human cell viability and mobility in hydrogel 3D bioprinted constructs. Here's some background about this project. Osteoarthritis is a progressive joint disorder estimated to affect 90 million adults in the United States in 2015. However, no disease-modifying osteoarthritis drugs are clinically available currently. 3D bioprinting is a novel tissue engineering technique impacting the field of cartilage regeneration that can hopefully develop a cure for osteoarthritis. We particularly define bioprinting as simultaneously depositing living cells and bioinks in a computer-controlled pattern. Bioinks that we use are JELMA and HAMA, which are photocrystallinkable bioinks that have shown biocompatibility in previous studies. Assessment of cell viability is essential to confirm biocompatibility of bioinks, bioprinting, and cross-linking methods that we use. Moreover, in this study, we'll assess the cell mobility, which is a complex process of cell translocation, extension, and orientation change, which is critical for wound healing. Understanding cell dynamics in bioprinted constructs is necessary to control tissue and extracellular matrix properties to create a zonally architectured cartilage construct. Moreover, there is a big gap in literature regarding the mobility of human chondrocytes and mesenchymal stromal cells in bioinks. Therefore, the goal of this project is to assess viability and mobility of human chondrocytes and mesenchymal stromal cells in selected bioinks. Now moving to the methods. The bioinks that we used are 1:1 and 2:1 ratios of JELMA-HAMA. We picked those bioinks because they differ in stiffness. However, in previous experiments, we've seen the similar amounts of collagen type 2 production. So we wanted to see if cellular mobility will be the underlying cause for this. For this project, we have performed 2 experiments. The first one was the life-death assay. For the life-death assay, we had 3 modal constructs. The first one was the printed construct with either 1:1 or 2:1 JELMA-HAMA that contained the cells. The second construct was the pipetted control with either bioink and the cells. And then finally, one more pipetted control with 1:1 JELMA-HAMA and the dead cells. This control was used to optimize the staining. All samples were cross-linked for 38 seconds with 405 nanometer laser. Further, the life-death staining was performed for days 0, 1, and 7 with calcine and ethidium homodimer, and the plates were imaged on the microscope, and the microscope software was used for quantification. The second experiment that we performed was cell mobility time-lapse. For this experiment, we had 3 constructs. The first construct was the sample construct, which is this 3-zonal construct. The first zone contains the cells. The second zone is the spacer, and the final zone contains chemoattractant, which is the fibroblast growth factor for chondrocytes and the platelet-derived growth factor for MSCs. The second construct that we used was the positive control construct, which had the seated cell suspension at the same concentration and printed chemoattractant section. Finally, we had a negative control construct, which has the section with the cells and double spacer. So no chemoattractant is present in this construct. All samples were cross-linked for 38 seconds with 405 nanometer laser. The mobility time lapse was imaged every 20 minutes for 20 hours. Further, all images that were produced were processed using FijiImageJ software, and mobility time lapses were further processed with TrackMate. Here you can see the results for the life-death assay. On figure 1A, you can see human chondrocytes and various bioinks for days 0, 1, and 7, as well as the quantification of those results. Human MSCs are displayed similarly in a figure 1B. Increasing viability of both human chondrocytes and MSCs is seen in both bioinks, peaking at around 100% by day 7. Moreover, the starting viabilities are no less than 80%, which indicates that bioinks, bioprinting methods, and cross-linking methods were biocompatible. Here you can see the results for the mobility time lapse. On rows 1 and 3 of this figure, you can see positive control for both human chondrocytes and MSCs. On rows 2 and 4 of this figure, you can see the human chondrocytes and hydrogel containing chemoattractant. Overall, the mobility of human chondrocytes and MSCs can be seen in this image. As you can see, those flagged cells are moving within the hydrogel. Moreover, the positive control samples show increasing cell mobility, which validates the design mobility assessment method. However, it also suggests that cells in hydrogels are a little bit less mobile. Direction and speed of cell movement have not yet been quantified. However, observation suggests that cells are directed towards chemoattractant when moving within hydrogel, as you can see right here. They're directed toward the source over here. And right here, they're directed towards this side as well. In conclusion, I'd like to say that bioinks and bioprinting technique were biocompatible with viability of more than 90% at day 7. Mobility time-lapse has shown cellular mobility within constructs. However, further analysis of cell tracking is needed to quantify direction and speed of cell movement. Moreover, further study could assess human cell mobility in hydrogels during chondrogenesis, composition of the produced extracellular matrix, and effects of varying hydrogel stiffness on cell morphology. Thank you for listening to my poster. I hope you enjoyed it.